Good morning, students. Today we are going to start Unit 2 Infrared Spectroscopy. In this PowerPoint, we are going to see the introduction of IR spectroscopy. Infrared spectrum is an important record which gives sufficient information about the structure of a compound. This technique provides a spectrum containing a large number of absorption bands from which a wealth of information can be derived about the structure of an organic compound. The absorption of infrared radiation causes the various bands in a molecule to stretch and bend with respect to one another. The most important region for organic chemists is 2.5 micron to 15 micron, in which molecular vibration can be detected and measured in an infrared spectrum and in a Raman spectrum. So, the IR spectra is produced by the absorption of energy by a molecule in the infrared region, and the transition occurs between vibrational levels. So, IR spectroscopy is also known as vibrational spectroscopy. Range of infrared radiation. The range in the electromagnetic spectrum extending from 12,500 to 50 centimeter inverse is commonly referred to as the infrared. This region is further divided into three sub-regions. One is near infrared. The region is from 12,500 to 4,000 centimeter inverse. Second one, infrared or ordinary IR. The region is from 4000 to 667 centimeter inverse. Third one is far infrared. The region is from 662 to 50 centimeter inverse. The absorption of infrared radiation can be expressed either in terms of wavelength or in wave number. Mostly, infrared spectra of organic compounds are plotted as percentage transmittance against wave number. The relationship between wavelength and wave number is as follows. We know that wave number equal to 1 by wavelength in centimeters. If the wavelength is 2.5 micron equal to 2.5 micron into 10 to the power of minus 4 centimeter. Then wave number is 1 divided by 2.5 into 10 to the power of minus 4 centimeter. That is 4000 centimeter inverse. The wavelength 15 micron corresponds to wave number equal to 667 centimeter inverse. In terms of wave number, the ordinary infrared region is 4000 to 667 centimeter inverse. You can see in the tabular column the region, wavelength, wave number and corresponding frequency ranges. Now let's see the principle of infrared spectroscopy. The absorption of infrared radiation causes an excitation of a molecule from a lower to the higher vibrational level. We know that each vibrational level is associated with a number of closely spaced rotational levels. So, the infrared spectra is considered as vibrational rotational spectra. All the bonds in a molecule are not capable of absorbing infrared energy, but only those bonds which are accompanied by change in dipole moment will absorb in the infrared region. Such vibrational transition which are accompanied by a change in the dipole moment of the molecule are called infrared active transition. So, these are responsible for absorption of energy in the infrared region. On the other hand, the vibrational transition which are not accompanied by change in dipole moment of the molecule are not directly observed, and these are infrared inactive. For example, Vibrational transition of C double bond O, NH, OH, etc. bands are accompanied by change in dipole moment and so absorb strongly in the infrared region. But transition in carbon-carbon bonds in symmetrical alkynes are not accompanied by change in dipole moment and hence do not absorb in the infrared region. Then, theory of molecular vibration. Absorption in the infrared region is due to the change in the vibrational and rotational levels. When radiation with the frequency less than 100 cm inverse are absorbed, molecular rotation takes place in the substance. So, as this absorption is quantized, discrete lines are formed in the spectrum due to the molecular rotation. Molecular vibrations are set in when more energetic radiation in the region 10 to the power 4 to 10 to the power 2 cm inverse are passed through the sample of the substance. The absorption causing molecular vibration is also quantized. So we can clearly understand a single vibrational energy change is accompanied by a large number of rotational energy changes. So the vibrational spectra appears as vibrational rotational bands. In the infrared spectroscopy, the absorbed energy brings about predominant changes in the vibrational energy, which depends upon the masses of the atom present in a molecule. Second one, the strength of the bonds and the arrangement of atoms within the molecule. It has been found that no two compounds except the enantiomers can have similar infrared spectra. Molecular vibration and origin of IR spectrum. There are two kinds of fundamental vibrations or stretching. In this type of vibration, the distance between the two atoms increases or decreases, but the atoms remains in the same bond axis. Second one is bending. In this type of vibration, the position of the atoms changes with respect to the original bond axis. We know that more energy is required to stretch a spring than that required to bend it. So we can say that stretching absorption of a bond appear at higher frequencies as compared to the bending absorption of the same bond. Various kinds of vibration. There are two types of stretching vibration. One is symmetric stretching. In this type, the movement of the atom with respect to a particular atom in a molecule is in the same direction. Second one, asymmetric stretching. In this vibration, one atom approaches the central atom while the other departs from it. There are four types of bending vibrations. First one is scissoring. In this type, two atoms approach each other. Second one, rocking. In this type, the movement of the atom takes place in the same direction. Third one, wagging. Two atoms move up and down the plane with respect to the central atom. Twisting. In this type, one of the atom moves above the plane while the other moves down the plane with respect to the central atom. Thank you.